Okay, so we've heard about the limitations of preclinical models for immune modulators and adoptive cell transfer and for vaccine therapies. And we also know that development of these immunotherapies really has varied from the development of traditional chemotherapy drugs, for example, and other anti-cancer therapies. And this creates challenges when trying to apply for INDs and for drug approvals, especially as these immune therapies become more personalized. So to touch on that, we have Whitney Helms, who is from the CDER branch of the FDA. Thank you.
Hi, good morning. As you said, I'm Whitney Helms. I'm one of the non-clinical supervisors in the Office of Hematology Oncology Products for the DOP2 team. And I'm going to be talking about the non-clinical data needed to support first-in-human clinical trials for immunotherapies. Here is the standard disclaimer that the views in this talk are my own and do not necessarily represent the views of the FDA, but hopefully they do. Okay. I'll have to start with the division of labor between the Center for Drug Evaluation and Research and the Center for Biological Evaluation and Research. So CDER does the monoclonal antibodies like pembrolizumab, nivolumab, ipilimumab, and some of the older ones like rituximab that may be considered immunotherapeutic. Also the fusion proteins like the bispecific T-cell engager blinatumomab, cytokines like IL-2, interferon gamma, and some of the antibody drug conjugates with a IL-2 or an IL-12 conjugated to an antibody. CBER, on the other hand, does the very exciting genetically modified T cells that we've been hearing a lot about today. The cancer vaccines like the Sipula cell T and the oncolytic vectors like the recently approved Imlygic from last year. So here are some of the regulatory guidances of interest when you're looking at your original first-in-human kind of studies. The International Conference on Harmonization, which recently changed its name to the International Council on Harmonization, has sort of the overarching guidances. These are developed across regulatory agencies with input from FDA, EMA, Japan, and some of your smaller regions like Brazil, Singapore, Korea, as well as pharma and FPA input. The one most relevant for cancer immunotherapies, at least from a CDER perspective, would be the non-clinical evaluation for anti-cancer pharmaceuticals, ICHS-9. They also have a Q&A document that's being developed right now, which may also be relevant. And the ICHS-6R1, which is the preclinical safety evaluation of biotechnology products, and its addendum. There are also FDA guidances. One that might be of particular interest would be the preclinical assessment of investigational cellular and gene therapies. So the ICHS-9 guidance tells you the typical requirements for the development of all anti-cancer drugs, and that would include cancer immunotherapies. To initiate clinical trials for anti-cancer therapeutics, you typically need 28-day GLP-compliant toxicology studies in 2 species. This is the traditional standard. But for biologics, as discussed in ICHS-6 and its addendum, a single pharmacologically relevant species is often acceptable. And that has been true for many of the antibodies, particularly because the rodent, it doesn't bind, so it's not really a relevant model. These are the studies that are the primary data used to determine the acceptability of first-in-human doses. So these are based on the severely toxic dose to 10% of the rodents or the highest non-severely toxic dose for a non-rodent. And in a lot of the anti-cancer pharmaceutical studies, mortality is your severely toxic endpoint. Other relevant endpoints might be CNS lesions that would be really difficult to monitor, or convulsions, gastric bleeding, those sorts of endpoints. And the goals of the non-clinical program are to identify the pharmacological properties of the pharmaceutical, to estimate a safe start dose, and to help understand the toxicological profile of the pharmaceutical so that you can do your relevant clinical monitoring. But one of the challenges we've seen with the cancer immunotherapeutics is that even more frequently with some of your more traditional biologics, like an anti-VEGF or an anti-EGFR, is that species relevance has really been an issue. So here's an example of a non-immune target, an anti-EGFR, as you might see in the Erbitux or Vectibix labels. You can see that the findings in the monkeys from these studies are actually very similar to what you see in humans. You see skin toxicity, often with secondary infections, eye redness, infusion reactions, mucosal toxicity. And these are all things that you pick up in the clinical trials and are part of the warnings for these products. But if you have the immune target anti-PD-1, and the ones that we've seen, while the affinity of binding to the human versus the cynomolgus monkey PD-1 has been pretty similar for all of these products, and the EC50s for blocking interactions have also been pretty similar. At exposures well above those seen clinically, there's no clear autoimmunity. Occasionally you'll see patterns of immune infiltrations into multiple organs, but it's very hard to translate that into real autoimmunity. And in the absence of knowing what the target really is, it's questionable whether you truly call that an immune infiltration that you would really note. This has long been a concern with immune targeting drugs. And ICH-S9 was written after the T. Gennaro incident in Europe. So they had special recognition of immune targeting drugs. So for biopharmaceuticals with immune agonistic properties, the selection of the start dose using a minimally anticipated biological effect level, or MABEL, should be considered. We've sort of taken this to also include a lot of the checkpoint inhibitors because you are still getting immune activation. And there's still concerns that you could cause unexpected effects. But determining a MABEL relies heavily on a variety of pharmacology studies, and it's not the traditional toxicology model that we've used for small molecules and even a lot of the biologics over the years. So it takes some getting used to, especially for reviewers who are more experienced with the small molecules. But there's no universal approach for determining a first-in-human dose based on a MABEL, regardless of the indication. Some of the useful inputs have been in vitro pharmacology data from target cells from human and toxicology species, concentration effect data from in vitro and in vivo studies, and if you're using animal data, then providing a comparison of species differences in drug exposure, target expression and distribution, and affinity of binding and intrinsic activity, as well as the dose-exposure relationship. We do look at the pharmacokinetics. So the things we're really looking for when we receive an immunotherapeutic in CDER are the pharmacology of the targeted pathway. Is it an agonist, or is it an antagonist, or is it an immune checkpoint inhibitor? The assessment of cytokine release potential, that's a pretty standard assay at this point. We see this for almost any immune modulating target that we get, and if you don't have this data, it might be a cause for clinical hold. Also studies using human cells that take into account multiple mechanisms of action, because these are the data that is often most useful in determining a MABEL. And also receptor occupancy, which I think can be a bit of a problem with some of these upregulated receptors. I think it can lead to really low doses, and we don't always really use that for making a MABEL determination. So here is the case example for anti-X. Target X is expressed on both activated immune cells and normal epithelial cells. So interactions between the target on the immune cells and the tumor cells are supposed to inhibit immune activation, resulting in tumor escape. So if you have the interaction, then you get a yield, but if you don't, then you're allowed to go. So the points to consider for this IND would be that the antibody was able to bind to activated human PBMCs, but there was no binding on monkey PBMCs, despite binding to the monkey protein by Biacore. So this called into question the relevance of the model even more than we typically see for immunotherapeutics. You don't know if the monkey is even having an immune response with this particular antigen. And the target wasn't very well described in the literature as something like a PD-1 either, so you don't have a lot of backing as to its immune-related activity. And expression is not restricted to immune cells. So if you're causing any sort of target damage and then you're inhibiting the immune response, you may be getting normal tissue damage that you're trying to avoid. So because of this uncertainty about the relevance of the monkey and the indirect immune activating potential, a MABEL approach was proposed. The sponsor used PK data from the monkey toxicology study to help estimate a predicted human exposure at a starting dose they proposed. And this was also based on in vitro data from interactions between epithelial cells and T cells in the presence or absence of this antibody. Another question, as you all know, is that what do we do with combinations? So traditionally, a lot of these combinations have come to us from research investigators with tons of clinical experience with the individual pieces, and those are usually dealt with reducing doses and proceeding based on the clinical experience. And in general, combination toxicology studies are not required. If you have a first-in-human study where you have no clinical experience with either product, then a combination study might be warranted if that's really the route you wanted to go. Typically, it's discouraged, partly because it would make it hard to pair it with other things, and most people do want to have that flexibility. What we will see occasionally is a sponsor will add a combination arm to one of their general toxicology studies if they want to start something very early. That's been one approach. But in cases where there's limited clinical experience, what we often will recommend are doing combination pharmacology studies, in vitro studies, or sometimes not xenograft, sometimes xenograft, but as you hear, there's some real problems with xenograft models with immunotherapeutics. Some of the interesting genetically, the mice with genetic tumors, those are kind of interesting models that we see sometimes. And very rarely humanized mice we'll see, but those are very difficult to make and maintain, and so there's not a lot of that coming in, at least to us. So here we have an example of two products with clinical experience which come in for a combination. So in this case, the sponsor proposed a combination of a kinase inhibitor with an anti-PD-1 antibody. There was significant clinical experience with both of the products. There were no pharmacology or toxicology studies submitted to support the combination. Occasionally, even if we don't have a study directly by a sponsor, we'll have something from the literature to help guide us, and we do find that useful in making these determinations. But in this case, we had an issue because we looked back at the original toxicology studies for the small molecule, and there were findings of severe cardiac inflammation in those studies for the kinase inhibitor. And while clinically there had been no cardiac findings noted to date, we had concerns that, despite the lack of clinical findings, there was concern about the possibility of exacerbating the finding and making it clinically significant by adding a checkpoint inhibitor. But despite this, there were no non-clinical studies requested. Instead, the sponsor was asked to lower the starting dose of the kinase inhibitor to a dose with a predicted exposure of about 20% of that causing cardiac inflammation in the non-clinical studies. So it was heavily reliant on the PK data from both humans and animals in the original toxicology studies. It becomes more complicated when you have drugs with no clinical experience. So we had a sponsor who submitted an IND for a first-in-human study using two products with no clinical information for either one of them. There was maybe experience with the class, but we are limited into how much we can use data from one product to really support the development of another product, both legally and because you have differences in antibodies. You're not making a biosimilar, so you can't really say that it's the same. And while we have to take into considerations safety findings from the class, it's hard to leverage that for activity and efficacy and unknown safety findings. So in this case, the IND was placed on partial clinical hold for the combination portion of the trial only. The clinical dosing had to be completed for each of the monotherapies in at least one cohort, and we also asked for in vitro pharmacology studies to help justify the combination dose. So when this one came back, the sponsor completed dosing of the first cohort for each product with no DLTs. And the proposal to use the first cohort for each product, the first cohort dose for each product, was still not fully supported by the limited experience and the ongoing worry about the potential for synergy. However, they also submitted some limited pharmacology data, and this was used to determine a reasonable dose in combination with the available clinical data. In this case, the product 1 dose was held constant while you increased the dose of product 2 and vice versa. And our concern was not picking a dose that gives you an exposure at the top of the curve. The experiments themselves were not the most robust, but there was a similar trend across multiple healthy volunteers. So a concentration for the product with maybe a little less experience was proposed down here lower on the curve, between an EC20 and an EC50 based on the totality of the data. And these types of decisions are discussed not just with the non-clinical team, but we really do collaborate with the clinical team so that it's a group decision. As opposed to some of your CMC holds, which might be for lack of viral clearance. There's not a lot of give in those sorts of holds. But with dosing, especially for things with clinical experience, clearly we're communicating with our clinical colleagues. And I tend to talk fast, so we're done a little early, but there have been a lot of limitations that have prevented reliance on traditional toxicology studies for predictions of safety for some of the immunotherapeutics. And while we're not completely ready to give up on the monkeys, if you do have an anti-PD-1 and you combine it with something else, occasionally we will see some sort of vasculitis or some clear effects. Or if you put in an antigen, you can sort of see some of the signs, maybe not as robust as in humans, but monkeys are able to respond. They just seem to have a different kind of threshold. And a thorough examination of the mechanism of the immune activity is critical throughout development, to inform not only the safety of the first in human trials, but also throughout development and into post-marketing. And that's really all I had today. These are my acknowledgments for Dr. Ricci, who had some of the slides that I borrowed, Dr. Layton, who is the division director for DHOT, and all of the DHOT reviewers. So thank you for your attention.